about fourth paradigm, etc. But here I put this in the context of how the mathematical modeling has developed over time, and how the data is converted to discovery over a period of time. So first, I share with you some of my own experiences, our own research, where we have used these three approaches to solve biomedical problems. Very long ago in 2004, we were looking at the data of DNA binding protein structures, and we wanted to know which of these proteins, if I just know the structure and if I do not know whether this is a DNA binding protein, what are the properties of the proteins I should look at. There were very complex models available at the time. I just try to summarize that, and I could identify that if we just count the total number of charged residues and total net charge on the protein, and thereby compute a rough value of a dipole moment and its quadrupole moment using certain approximations from even low resolution structures of proteins, we could predict DNA binding proteins from structures with high degree of accuracy. This was done way back in 2004. And this is where the quality of data comes into play. Here, the model itself is not so sophisticated. Models are simple, but the data that goes into these models are of a very high value or of a very high quality. And then only the simple models can emerge from the data. Otherwise, all it will produce will be noise, and no modeling can be carried out. So that is where these models become successful. This is the older, the traditional, the first paradigm method of looking at the data and making discovery. More recently, in 2014, another high quality data we compiled from Protein Data Bank, and we curated data of DNA binding proteins undergoing conformational changes. When they bind to DNA, their structure changes. This is very important because if we create a homology model or any low resolution structure, we will probably not know how the protein will actually be when it binds to DNA. And a lot of conformational change takes place in this process. So if we classified these conformational changes into 6 categories, and each of these categories had a distinct signature in terms of the stability and the specificity, for example. And this helped us to make discoveries about these relationships and also predict what will happen to the complex. Will the complex be stable? Will the complex will be weakly stable, and etc. These properties could be related to the conformational change categories that we identified in 2014. So this was the first paradigm, a simple overview of how the simple things can lead to interesting discoveries. Then we come to the second paradigm, because the simple statistical models, where we just collect high quality data and we try to model them, we are trying to make some very simplistic assumptions that, such as linearity, non-additivity, like x plus y is equal to xy, or there is something that x plus y is more than x plus y. That is, so this is the non-additive behavior. That is, if you, for instance, if you take a practical biological example, it will come, I'll show you in the next slides, that if you remove one residue and the energy lost is E1, and if you lose another residue and the energy loss is E2, if I remove both of them, the energy loss is not E1 plus E2. It is either more than E1 plus E2 or less than E1 plus E2. So the residues are interacting with each other. In the context of a computational model, if I want to predict something from a set of features, then these features are interacting with each other. So there is an inherent complexity in my data. So the simple models of statistics, and such as linear regression or simple curve fitting, will not be able to solve these problems. We have to find models which can take cognizance of the complexity of the relationships between the features that we use as the input, for instance, a set of genes, expression values, and the output target properties that we want to see whether a disease is likely to be encoded in this gene expression profile. So this is where the machine learning comes. And we early on, when we were trying to predict the DNA binding residues, whether there's a whole sequence of DNA binding protein, and we want to know which of these residues in the protein sequence actually interact with DNA. So this does not happen in a non-additive way. Every residue participates in interaction with DNA, not on its own, but in cooperation with the surrounding residues. But the surrounding residues do not join or combine with the key residue in a non-additive way. Just again, example, E1 plus E2 is not just an algebraic sum when E1, 2 takes place. So this kind of a non-additive behavior If we want to introduce it into the simpler model, such as if we want to create a multiple regression model of the interacting pair of features, then the complexity becomes too high. Traditional models, statistical models cannot handle that. So if I want to incorporate a polynomial where the features can be added as pairs, so the number of parameters that my model requires to train or optimize is n square. So this complexity becomes very, very high. And any model which is based on this high complexity is overfitted and does not have a practical value in the application stage. So that is where the machine learning, particularly neural networks, become very powerful. Neural networks can model very complex surface between the feature space and the target space with very fewer parameters than, say, multiple linear regressions where you have complex features included, or Bayesian models where we want to include the pairs of residues, one dependence models or multiple dependence models. If we want to incorporate the feature interaction into these models, they become very complex. The neural network can model very arbitrary, very difficult, very complex surfaces with much fewer number of parameters, and therefore can avoid overfitting. So that is where the classical machine learning helped, and these are some of the papers we published in this field to predict DNA binding residues with the help of neural networks. From 2004 onwards, in 2009 was this was the main area of activity for my research. Going further on, we were also looking at the residues, amino acid residues in protein sequences. And as I said, that the different groups of DNA binding proteins, the nature of evolutionary profiles, the conservation of residues was found to be different. And again, we could predict with the help of this, if I can identify a cluster of conserved residue, I can tell this is the cluster of conserved residue which is crucial for binding to DNA or interacting with the genome, and as a transcription factor, initiating the gene expression process. So these two papers we published in Nucleic Acid Research in 2008 and 10, both of them basically talked about this idea of the clusters of conserved residue. The technology here, as you see here, the clustering technology, the machine learning, the unsupervised machine learning, where we do not have a target property, we are just looking at how the charged residues come together and form clusters, and then we just find these modules or these clusters within the protein structure and then characterize them. And then this will be, so in combination of the unsupervised learning in the form of clusters and then subsequent domain analysis of the looking at the biological properties and the thermodynamic data of protein DNA interactions, we were able to identify some very unravel some very interesting properties and show that they interact in a cooperative manner. So now coming back to the third stage of the current paradigm of pattern mining, I take some time to spend on what is happening today in our lab, particularly in the context of my own research. So first topic, the issue that we are dealing with is the in search of DNA recognition code in cellular contexts. There are many questions about how the transcription factors regulate certain genes. Many questions are still unanswered. For instance, there is a transcription factor, and in the entire genome, there are many occurrences of the same sequence motif. This sequence motif is recognized by this transcription factor in some places, but it is not recognized in some other places. So out of the thousands of genes which have a given target sequence in the promoter region for the same transcription factor, some are regulated in one cell, some are regulated in another cell. So we do not know what causes these particular genes expressing in one cell and some others expressing in another cell, although transcription factor is available in both cases. So this is one primary question. Another question is that the genome is so long, and if the transcription factor has to find where it has to bind, it takes a very long time for transcription factor to scan. But transcription factor can actually quickly find its location where it has to bind. So how does the transcription factor go from completely cellular distinct area like a cellular region and go to the nucleus and then identify where it has to bind? But even more intriguing is that the backbone of the protein, the backbone of this DNA is identical everywhere. the bases which determine the sequence of the genome are where the are actually not visible to the transcription factor.